Good morning. Today you're going to get two for the price of one, so there's two of us speaking today. I'm Richard Salvage from Maxtim and Queen's University in Belfast, and I've got Dr. Colin Fleming from the Agri-Food Bioscience Institute and Queen's University to help me out as well. So we're going to talk about biostimulants. I think there's a lot of interest in biostimulants, increasingly so, and how they can be used for managing stress in sports turf. So one of the first questions is, what is a biostimulant? We'll be talking about that. Why would you use them? Increasingly important, how do they work? A lot of people ask, do they work systemically? What actually is the mode of action? The answer is very complex, but we're going to attempt to do that. And then how can they help you manage stress? Now, there are well over 50 published definitions of a biostimulant. This is only one of them. And this is from the European Biostimulants Industry Council. And they've defined a biostimulant as a substance or substances and or microorganisms whose function, when applied to plants or the rhizosphere, is to stimulate the natural processes, either to enhance or benefit nutritional uptake, efficiency, tolerance to abiotic stress, and crop yield and quality. So, biostimulants distinguish themselves really in two ways from normal inputs. And they've got very different mechanisms to fertilizers, regardless of any nutrient present. And they also differ from crop protection products because they act on the plant's vigor and health rather than act directly against pests and diseases. So, obviously biostimulants are used in agriculture as well as sports turf, and they're basically biologically derived and can be applied alongside fertilizers. They're not food, essentially. But the purpose really is to help improve the nutrient use efficiency. They help plants tolerate abiotic stresses such as heat, cold, drought, high salinity, etc. But they also improve quality attributes like appearance, color, rigidity, and nutritional content. And we also see protein increases as well in grasses. Now, more and more biostimulants are coming available, and they're coming from essentially three main sources. There's acids, organic acids like humic, fulvic, and obviously amino. The microbials, so mycorrhizae, trichoderma, and also plant growth promoting bacteria. And extracts. A lot of you be aware of seaweed, chitin, and other plant extracts as well. And then there's one or two other products which fit into the scheme of things. But all of these are being worked on and developed, and we see a lot of activity. So, biostimulants, they certainly foster plant growth and development, but they do it through the whole of the life cycle. And we've certainly seen when certain biostimulants are applied as early as possible in the life cycle of a plant, that they do have a strong effect. So even from seed through right through to plant maturity, we see improvement in efficiency of the plant's metabolism, which can increase yield and develop quality. We also see an increase in plant tolerance to stress and recovery from stress. Better nutrition use and increase in bricks, carbohydrates, the color, chlorophyll, and we see plants using water more efficiently too. And they can also have a big impact on the quality of the soil and the microbes within it. One of the things that many of you will have noticed is that more and more crop protection products are being discontinued. There's a EU policy really directed towards significantly reducing the availability and usage of agrochemicals. So what do we use in their place? A lot of countries already have action plans in place to reduce agrochemicals. I'm certainly aware in Holland of an attempt to reduce to 50% in agriculture. And that's a big change. That is a very big change, and it's going to affect all of us. So, what are available? Looking at the different categories, the vast majority are sort of what I call single source biostimulants. So you can obtain seaweed extracts, you can buy amino acids, humic acids, etc., etc. And all of these are available as a sort of single source biostimulant. There are a few more complex or multi-source products where they use a blend of these single source to create a synergistic effect, and Maxtim fits into this category. And then there's the microbial. So we've got the fungus, the mycorrhizae, trichoderma, and the bacteria. And what we've been playing with recently is combining some of the microbials with Maxtim. We've been sourcing our mycorrhizae from PlantWorks, and we've seen a synergistic effect. And really it sort of leads to a fourth category, even more complex, mixing some of the complex chemistries with the microbials. So I just wanted to show you, just give you a taste of what we've seen when we do mix them. So what we've got here is an analysis of root mass, starting with the root length. On the left, we've got the control. In the middle is the arbuscular mycorrhizae fungus that we've used. And you can see there's a significant uplift in root length. But when you apply that in conjunction with a Maxtim formulation, we see a synergistic uplift. But we see that also in the root mass. So the number of splits, the tips and forks, again, a significant uplift and a synergistic response between complex biological sourced chemistry and microbials. Okay, what is Maxtim? Well, basically, it's a very complex blend of some of those single source components. We use a carbohydrate matrix into which we suspend some specialized plant extracts, organic acids, antioxidants, amino acids, a whole selection of products. And we see this synergistic effect because of that. But the science behind how they work is in its infancy, really. And it's something that we've been working hard on to try and illuminate some of the modes of action which are going on. Hugely complex, massively complex, and it's going to be a lifetime of research ahead. But we want to understand better how they work. Now, we've got a massive and intensive in-house R&D project going on all the time, but it's important that we also collaborate with centres of excellence. So we're working very closely with the Agri-Food Bioscience Institute in Belfast, the Institute for Global Food Security at Queen's University, and the Institute of Life Science at Swansea. So I'm going to ask Colin now to talk about some of the experiments that we've been conducting, and we can discuss some of the results that we found.
Okay, thank you. As Richard pointed out, understanding how biostimulants work, I think, is crucial to their effective use. If we know how they work, we can target specific biostimulants and formulations for particular problems. At the moment, we're sort of just shotgun approach, hoping that something will work. The more we know about how they actually affect plants, the better will be our usage of these things in the future. And my research group in Queen's and at AFBI really is a merger of innovative commercial companies like MaxStim, scientists like myself, but also, and maybe most importantly, greenkeepers and groundsmen like yourselves. All three groups have to work together to develop a better understanding of biostimulants. So what I want to do is to show you some of the research we're doing, and it's helping us understand how these things actually work on plants, and from that, how we can use them more effectively. As Richard pointed out, roots are one of the key changes we see in plants when biostimulants are used. And the sort of work we're doing is we're treating plants, we're taking the roots, staining them, and the dyes we use are taken up by the root. That means we can actually see the roots much more effectively. And this shows you some of the stained roots we're working on here. And we can scan these using specialized software. And the scanner allows us to measure the full root length, the diameter of the roots, the number of branches, and the number of root tips. And this is all crucial to understanding how roots change when treated by biostimulants. And just to show you some of the data, this is root length. This is actually on ryegrass, Lolium perenne, and you can see there's a significant increase in root length. We also see major changes in root morphology, and you can see here the number of root tips increases, as do the number of forks. So we're seeing massive changes in the root structure. In some cases, this will be beneficial, but in other cases, there could be issues. And that's why we need to understand the underlying biochemistry and genetics behind these changes. We're looking at lots of different types of stress. We're looking at drought stress, cold stress, heat stress, even high salinity levels. The example I'm giving here is drought stress. It's probably one of the most important in turf management and in crops. And drought stress affects a wide range of things inside plants. And many of the negative physiological effects you see from other stresses, such as heat, UV light, cold, freezing, they're all down to osmotic stress. So this is a big interest for us. And whenever plants are experiencing a stress, massive changes are going on inside the plant. We're seeing changes in the transpiration, the rate of water movement through the plant. We're seeing buildup of osmolytes in cells. We're seeing changes in root structure. We're seeing antioxidants being produced. All of these are going on in the cells, and it's a very massively complex situation. But basic to all of this is molecular response. All of these changes are being governed by gene regulation and gene expression in the plant. The plant is changing how it works, its genes, and its protein production, all to allow it to manage stress. And the key thing is, can we actually help the plant by modifying these things through the use of biostimulants? One of the really interesting new concepts in biology is defense priming in plants. It's known as plant memory. And some people say, hey, plants can't have a memory, but they actually can. And priming is the method they use to basically prepare for a stress that may appear downstream. It's a bit like us taking a vaccine. It protects us against a future disease, and plants do exactly the same thing. And this chemical priming allows us to prepare a plant for a stress, and that stress can be abiotic, for example, heat stress, drought stress, or it could be a stress produced by a fungal attack or an insect attack. And this concept of priming, I think in the next 20 years is going to revolutionize crop production and also turf management. So how does it actually work? What you do, you give the plant a minor stress, and this could be a biostimulant, it could be another chemical, and it basically imposes a small stress in the plant, and this changes the gene expression in the plant. And this stress priming period can last for days to weeks. What happens then if you actually impose a major stress, so say your green is subject to very high temperatures, the plant immediately responds with a defense response very, very quickly. You can see here, here's the untreated plant, so a green that hasn't been pre-treated, and it takes a much longer period of time to prepare that for that stress that has hit it. And it's a bit like hardening, you take your plants out of the glass house in stages, exactly the same principle. And the questions we have now are, number one, how do biostimulants work to protect the plant from stress immediately? Can we use biostimulants to actually help the plant recover during stress? So if your green is being hit very hard, can you apply something to help it recover immediately? And thirdly, can we induce this priming response? And a lot of our experiments at the moment are designed to try and answer these questions. And what we're trying to do is to provide you guys with new tools to manage your turf in different situations. So one particular experiment we're doing is we are taking plants here and we're basically stopping watering. We're hitting them with a drought stress, and at the same time we're spraying with biostimulant. And what we want to know is, will this spraying protect the plant as it undergoes this stress? Second experiment we've got is where we are imposing a drought stress on the plant, and then whenever the drought stress is well established, we're spraying them. We want to see if this spray will help the plant recover from the drought stress. And the final thing we're trying to find out is, can we induce this genetic plant memory? Can we actually prime the plant? And how we do that is we spray the plant, we continue watering, and then a week later we hit them with a drought stress. We want to know, will that plant be able to recover from this stress better than an untreated plant? So that's the type of experiments that we're actually setting up. And, sorry? You're going to see the results now. And this is one of them. So in this case, what we've done, we have sprayed the plant as we induce a drought stress. And you can see here all our different treatments. There are no real differences. So this biostimulant has not helped protect the plant at all. The treated plants are no better than the drought stress plants. The plants eventually die. However, look here. In this case, we've taken a seaweed extract and we've pre-treated, and we've let the plant survive in good growing conditions for a week. And then after 7 days, we hit them with a drought stress. So here's our droughted plant control, untreated, and you can see the plants are all dying off. Look at this one. This has been pre-treated, and what we're seeing here is evidence of genetic priming. The plant basically has been inoculated to survive this abiotic stress. And when we look at the results, here is the control plant, the plant that has been watered during the experiment. You can see that our seaweed extract plants are doing as well as the control. These are the droughted plants down here. So we've got now evidence that these seaweed extracts can actually prime the plant to survive a drought stress. The other question we want to know is, can we actually protect the plant when it's undergoing stress? And what we've found here with this particular biostimulant is that here's our plant. We have induced a drought stress, and halfway through the drought stress, we spray them. And in this particular treatment, you can see the plant has recovered. So what we've done, we've brought these plants back from the death. All the other plants, including the control, die off. This plant has recovered through the use of a biostimulant. This is the approach we're using now to try and identify how we can use biostimulants more effectively. This is that previous experiment. Here's our control. And you can see that the treated plants, which have undergone drought stress, are doing as well as the control. And that's through simply recovery by using a biostimulant. Now, understanding the genetics of this is our next step. And we're using a technique called RNA sequencing. And RNA sequencing allows us to look inside the plant cell and determine exactly what genes are being changed by the biostimulant. And we use some very complex chemistry and machines. But basically what we can do, we can identify what genes are being turned on, what genes are being shut off, and what genes aren't changing. And this is just one set of data. Here we've got plants treated with salicylic acid, which is a plant hormone. Basically, it's aspirin. It's found inside all plants. It's a defense hormone used to protect the plant against fungi and also drought stress. This is methyl jasmonate, protects the plant against insect attack, but also, again, effective in abiotic stress. And this is seaweed extract. And what we've got here are the numbers of genes that are basically turned on by the treatment. So salicylic acid, over 600 genes in that plant have been switched on. So it's having a massive effect on the cellular processes inside that plant. And you can see here the seaweed extract is also increasing. But what's really interesting are the genes in here. They're turned on by jasmonate, by salicylic acid, and the seaweed extract. So these 53 genes are the really ones we're interested in. They're the genes that are probably involved in allowing the plant to survive stress. And when we actually look at these things, we can see the genes that are being turned on by the biostimulants. So it's chlorophyll production, it's genes that are involved in protecting the plants against damage and cold stress, defense genes, genes involved in upregulating general activity in the plant, and genes involved in drought stress as well. So we now know the genes that are being turned on by these biostimulants, and we can now predict when we apply a certain biostimulant what well may happen in the plant. And that then allows us to design new biostimulants specifically for certain types of stress, be it salt level, could be drought stress, cold stress. And the development of these new technologies are going to allow us to actually apply biostimulants much more effectively in the field. And I think Richard now will show you some examples from case studies where we've used these new biostimulants in the field, hopefully to get a reasonable response.
Thanks, Colin. Okay, the first one I want to show is Dunmurry Golf Course. 95% power, and it was subjected to a maximum treatment over 12 months. We kept one green as a control, and we monitored root development as one of the key parameters. Obviously, you're all aware that the poor varieties, it tends to have a shallow root, can be very stressed in summer, and any root development you can get is going to help. So this is what we found. The control green here, the chipping green, we saw obviously root development over the 12 months, but compared to the root development on all the other greens, there's a significant improvement throughout the whole course. Even on these two, where everybody's got a couple of shaded greens, and indeed these were the shaded ones, but they've developed strong root growth throughout the 12 months. But we also saw virtually no signs of stress or disease throughout the course, except on the untreated. It was subject to stress. And this is where Colin spends most of his time, apparently, getting his handicap down. Is that right? We did an extensive trial on the Golf National in Paris. We've got a small event coming up this year. They've been using Maxim for a couple of years, but when we started to evaluate how we could help them, we did a massive survey of the root system on the Eagle course, where we took 3 cores in every green, so we could evaluate the root condition before we set the treatment up. They had 3 treatments of Maxim, and then we took another 60 cores and compared, and we saw on average a 36% increase in root length. But we also saw increased chlorophyll and an overall improvement in plant health. So this was a typical example of before and after. And Alejandro's very happy, and he's been using it now for a couple of years. As I said earlier, we see big changes at various points in the plant life cycle. Anthony Harries at Swansea City has been experimenting with using Maxim at seeding, and in fact sprays the Korodov pitches just prior to seeding. And he sees early germination, really strong, healthy plants, and goes from seed to that in 14 days. Now, some stadia have a big pressure on time, because not only do they use for football and rugby, but sometimes there are concerts to recover from as well. And so the growing period between seasons is getting a bit tighter. We did some work at Chantilly Racecourse. Some of you might know that they've had the Prix de l'Arc de Triomphe here for the last 2 years while Longchamp has been renovated. We did a trial on a hectare of the track and compared it to the untreated. And essentially, the results were extremely visual. We got nice white root, young root growth, whereas we were getting nothing from the compacted substrate in the untreated area. So, and that was only after 2 treatments. Now, it's not a cure-all. It doesn't replace good practice, and everyone's here to get better at what they do, but Maxim's here to help. But we know the more we look at it, the more we know we don't understand. So it's going to be a lifetime of work to really understand how some of these biostimulants work. But we are committed to keep on designing and looking and trying to understand, and we're happy to share the information with you. Well, that's it. Thanks very much. Thanks for coming. Fill the room. Happy to answer any questions. Hi.
Question for Colin. See the priming sort of side of it. If you were on a monthly application, anyway, say with the CV product, would that be the same as a priming plan, or is there another way?
Yeah, the question is, if you're on a monthly application of biostimulants, is that inducing priming? And the answer is yes. That's why I would advise guys to use biostimulants fairly frequently and surprisingly at low concentrations. You don't need to overdo it. And what you're effectively doing is preparing the plant, changing the gene expression to allow it to respond quickly to whatever stress may hit it. And it's interesting that this priming, this memory, genetic memory, was first identified in humans. And it was guys that had been in concentration camps during the war. And they found that they were undergoing severe stress, and they could measure the stress in their physiology. And what they found was the children of those guys showed the same stress levels. So this is a memory being passed down the generation. And if we grow plants, induce a drought stress, the F1, the seeds, are also protected because this genetic memory is passed on through the generations. So if you've got Poa annua, or an inducing basic resistance to a stress, the seeds produced that year should also be resistant to the stress. That's the theory. Haven't seen that in practice yet. But little and often, yeah.
Your product is very expensive. And you've said yourselves that you don't know all that it does or how it does it. Why shouldn't I go and buy 200 litres of complex carbohydrate, which is really just
Molasses.
Down at the farm shop, mix it with a bit of seaweed, maybe put a touch of urea in it, or something like that, and get exactly the same response for a tenth of the price?
Well, you won't get the same response. We've looked at, you'll get a response, but we've looked at every component and we've optimized the concentration of each component. And it's not, we use the molasses as the matrix.
Carbohydrate.
Yeah, as the,
It's molasses.
yeah, as the suspension medium.
Yeah.
If you took it away, you would still see a significant impact. But when the components are used individually, you don't see the same effect as when you combine them in the precise blend that we have. Most of the work that we do we normally compare against the current practice or the current best practice. So it tends not to be a totally untreated. I mean, you're always going to get a better response against a totally untreated, but that's not necessarily valid. So we always look at comparing to the current practice or current best practice.
So all designs are taken in by the plant, is it no soil application for the
The question is, are there differences in foliar uptake and root uptake? Yeah. The answer is yes, and that's a part of the reason why we are investigating different components of these complex mixtures, because some components are obviously taken up foliar, others are taken up by the roots, and others actually affect the microflora around the root. So it's gaining a better understanding of exactly what each component is targeting in and around the plant that will lead to more effective use and better designed biostimulants. I think that the question earlier regarding molasses and complex mixtures is very, very important, because for 30, 40, 50 years I have known greenkeepers using all sorts of mixtures and getting good results in some cases. The question is why in some cases did it not work? And it's through understanding the processes that are going on inside the plant that will allow us to avoid those mistakes and those failures. And that's increasingly becoming very important as we're putting our greens and our pitches under a lot more stress.
Just a question in relation to biostimulants and their relationship with plant growth regulators. Is there a contrast or do they contradict one another?
Yeah, the question is, will plant growth regulators and biostimulants be antagonistic? And the answer is, in certain cases, they can be. The problem is, again, in, for example, things like seaweed extracts, we see some components in the extract enhancing growth, others actually retarding growth. And it's understanding which ones improve growth or reduce growth are important. And other components will also react differently with plant growth regulators. So it's a very complex situation. I don't know the answer to every single interaction, but that's something we're working on to try and understand better how different materials that are put onto pitches and onto greens work together. And that I think, again, is going to be a very difficult question to answer. It's going to take a lot of work.
Hello?
Yeah, the question is, will priming of the grass plants increase seed production? The answer is actually fairly complex. When we look at the genes that are involved in the priming process, we're seeing literally hundreds of genes are being affected. Some are being turned off, some are being turned on. One of the genes we see is involved in seed production. It's actually flowering in seed production. So the answer is, certainly in some of our experiments, we see an increase in seed production, which again affects the physiology of the plant in a different way. So you can see it's a very, very complex situation, and it's going to take more than a lifetime, I think, to work out exactly what goes on with every possible combination. But the more research we get, the better understanding we have, and therefore I think the better the tools we can provide you guys in the future.
The case studies that you did, was it just with MaxStim, or was it the MaxStim and the mix with a microbial product as well?
The first set of graphs I showed you were with the mycorrhizae, but those case studies were just MaxStim.
Some of the experimental work I showed you were other biostimulants, not just MaxStim. I think the key thing is identifying what are the really important components of biostimulants, because if you look at there are literally thousands of mixtures out there. It's understanding which components are key to better management of turf and stress.
All right, well, thanks very much. That was a good session. Hope you enjoy the rest of the show. If you want to know more about MaxStim, come and see us at the stand. Thanks.